I'm now going to talk about a very, I'm just going to now talk about something related to sequencing and algorithms, because I think it's interesting, even though some of you might have seen this before, because if I've talked in some of your other classes. And we're not actually going to cover this until after the midterm, so maybe a month from now. But I think it's just a good idea. We're going to go through this and think through this, because it just gives you a flavor for the kinds of things that we're going to be covering in the class. Okay, so let's say I want my DNA. So there's me, and I want my DNA sequence. This is my DNA sequence. Okay, this is a 3 billion length sequence. Okay, and it's conveniently annotated in red letters where all the polymorphisms are. Right, so that after I have that, I can readily include myself in a disease study. Okay, so that's what I want. But obviously, it's not like you need some kind of, like if I spit or something like that, there's DNA molecules in my spit. So how do I convert those molecules into a sequence? Okay, so we use something called a sequencer. This is actually called this is a Illumina, called Illumina sequencer. This is a picture of it. They had the unfortunate kind of idea of calling it next generation sequencing. This is like 5 years ago. And so now they have new ones. They don't really know what to call them. It's actually really funny. But it's called next generation sequencing. And so it's very cost effective. That's why the prices are so cheap. But the thing is that they give what's called short reads. Okay, that's like why they're cheap. They use this technology called short reads. What are reads? So this is actually how a sequencer works. Okay, so this is kind of a way that the sequencer works. So imagine that there's like this full DNA sequence that's encoded in that molecule of DNA. Okay, I don't have it. Okay, but it's just, it's in the molecule. The way a sequencer works is that you could think of the sequencer as basically breaking up the DNA into like millions of little pieces, okay, and just outputting the sequence of those little pieces. Okay, so you could think of it just like a model for how the sequencer works is that it picks a number from 1 to 3 billion, okay, and then just outputs the next 30 positions. So you get these what are called reads, okay. So these are like length 30 reads that are just randomly drawn from this long sequence. Okay, this is called short read technology because these reads are relatively short, so like 30 or 100 or 200. While the previous technology, the one that built the human genome, used reads that were like 10,000. Okay, so these are much shorter, but they're shorter. They're cheap because they're shorter. Okay, essentially. Okay, so the reads themselves don't have any information on what part of the genome they came from. You just get the sequence. So the question is we don't know where each read comes from. Okay, so we just get this long list of reads, and we can't identify where the mutations are because it's not like I have any kind of coordinate system on these reads, and so the question is, what do we do? Okay, so any ideas? Yeah. Yeah, so that's a great idea. Okay, so and I know that many of you have already seen this, right? Probably, but you forget, right? Those of you that seen this, you forget. Humor me, right? Okay, so you forget. Okay, good. I don't feel as bad. So one way to do it is look at overlaps. Okay, so what you do is you take, let's say these are length 30. Okay, let's say 2 reads overlap by 10. So then what you could do is you could just say, oh, well, they might have come from the same place, and now you have a read of length 50. Okay, and then you overlap another one, and you get a read of length 70, right? And then 90, and then you just keep on repeating that process until you get to 3 billion. Okay, that's one way. That's called sequence assembly. That's a great idea. That's actually how they assembled the original human genome. There's a problem, though. What's the problem going to be? Yeah.
Probably going to overlap with
Yes. Yeah, yeah. It might overlap with a lot of different places. Okay, and so how many places will it overlap on average? If you're using reads of like overlaps, you're asking for 10. Okay, so let's do the math. So let's even pretend the human genome is a random sequence. Okay, just to make our lives easier. Okay, so first of all, how many? So let's think about this. How many? So how many possible sequences are there of length 10? 4 to the 10th. Did someone say that? Right? Okay. So what is 4 to the 10th? Huh? 2. Yeah. 2 to the 20. Yeah. Yeah. And how much is that? Huh? A million. Yeah. Very good. How do you guys should be able to do that like in your head? Okay. So I'll tell you why. Okay. So 4 to the 10th is 2 to the 20, right? 2 to the 20 is 2 to the 10, right? Times 2 to the 10, right? 2 to the 10 squared, right? So what's 2 to the 10? Like 1,024, right? So 1,024 squared, 1,000 squared is about a million, so it's about a million. So that way, like, you see, like like knowing tricks like that is how you impress people, you know? It doesn't matter if you know like a lot of other things. If you know tricks like that, they'll be like, wow. You really are good computer scientists, yeah. And so for those of you that are in biology, this is even more important for you, you know, these kind of things. Okay, so there's on average there's a million. So then if I have a sequence of length 3 billion, how many on average? How many times will every sequence of length 10 occur? This one's easier actually. Huh? Yeah, 3,000. Yeah, that's because 3 billion divided by a million is 3,000. Okay, so even if the genome is random, okay, you'll see on average 3,000 other positions that match in the genome. Okay, and so then you might get it right when you pick. You'll have all these different options where to overlap. You might be right, but there's also 2,999 chances that you won't be right. Okay, so what will happen is it'll be really ambiguous. This is actually related to a problem called sequence assembly, and we'll talk about that later in the class. And that's actually a possible class project on how to do that. Okay, so that's not going to work. What's another strategy? Any ideas? So you can collect more data, right? That's always usually a great strategy. In this case, it won't help you because you'll end up with even more overlaps. So, you can get. No. So even in that, I see what you're saying. You still won't. You're saying that if you get even more data, then you'll get overlaps of length like 29, like out of 30, huh? 20, right? Right. So that's a great idea. So it turns out that in a random sequence that might work. Okay. But the human genome actually is extremely repetitive and has lots of regions that are like exactly the same, that are much longer than 30. So it's not going to work. So, yeah. So any more ideas? There's kind of a pretty, you can use longer reads, sure, but that costs you more money. So nobody wants to do that. So what, yeah? Yeah, so you can compare to the reference. So the idea is that we're not the first ones trying to get the human genome. There's already a human genome sequence that's out there, and this idea is that you can use resequencing. It's called resequencing or read mapping. So the key idea is that we know that the genome is my genome is very close to the human genome. I only differ by 0.1%. Okay, well maybe me, because I'm kind of weird, 0.2%. Okay, but most people 0.1%. Okay, so. What do you do? So you take, this is my genome. Let's say I get a sequence read. This is a random substring from my genome. Actually happens to start here, okay? And then what I do is I go to the human genome and I try to find where it matches most closely, okay? And you see it matches here. So that means that it probably came from here, right? And then what I do is I look at the places that differ between the human genome and my genome, okay? And I record a list of those differences, okay? For every read, I just keep on adding to this list. Does anyone ever use diff here on Unix? No? Does that sound familiar? So diff, what diff does, it takes two documents and gives you the list of differences. So this is kind of like, you can think of this list as being the difference between my genome and the human genome, okay? At the end, I get this list of differences, and what I do is I just take the human genome, the original human genome sequence, and I just add all the differences to it, and it'll be like my genome. Okay? Yeah.
Won't that subsequence match on the human genome several places?
Yeah, that's one possible problem. Yeah. So we'll get to limitations of this in a second. That's a great question. Okay, so that it can be ambiguous, but this is the basic idea. Okay, so so this is the list of changes, and then so this is kind of similar to a diff. Okay, that's the idea. Well, here's actually exactly what you asked. So what if you have a region that's repeated here? Okay. In the human genome, you have two regions that look exactly the same, and I get a read from this region. In my genome, one of my regions that are repeated has a difference, has a mutation there. Okay, so I have a read from here. When I match it, it'll match into two different places. Okay, and so my recovered sequence will want to put this difference in two different places. Okay, so that'll be an error. Okay, so again, this is a limitation of this approach, is that if the read doesn't match uniquely, then I don't really know what to do. Okay, that's exactly your question. That makes sense? So, okay, here's another type of error. What happens if, so actually, well, what'll happen if my genome has a lot of genetic variants all next to each other? What'll happen? Huh? Yeah, so it won't be close. So for example, here's an example. Here's my genome. I have all of these mutations right next to each other. Here's a read. So in the human genome, it doesn't match here, right? So there's no way I can know that it came from there, right? That's another example. So if there's too many, then I can't identify where the sequence is. Okay, so the question is, when does resequencing work? So we must be able to map a substring from the target to its corresponding place in the genome. And when can this happen? So if there's repeated sequences, and if there are differences, or another reason why it cannot happen is if the target sequence differs, differs a resemblance to the sequences actually. So these are reasons it cannot happen. Okay, so actually I'm going to skip this. Okay, so okay, but anyways, let's ignore those problems, because it still works most of the time, and let's talk about why is resequencing hard. So why is resequencing hard? Any ideas? Why is this a hard problem? Yeah. Oh no. Slow. Yeah, it's slow. How slow? So what makes it hard? So the genome is 3 billion. Okay. Sequencers generated maybe 300 million reads or more, right? We need efficient algorithms to map it's called mapping, each read to its location in the genome. Okay, and so there are other challenges we're not talking about, but just like here's what's the most trivial approach to map to find where it matched in the genome, where a read most closely matches. Yeah. Yeah, run through the entire genome. Okay, so here's a read that comes from there, and we go from we slide our read along the genome, and we just count how many times it matches. So here's it going at the first place as a match. No, these are all the places it mismatches. So it doesn't match, right? So then I slide it one more. Okay, does it match? No. Does it match? No. Okay, I think it decided that it's okay there. So I skipped because I got bored. Okay, does it match? No? Okay, there, finally, it matches, right? So how long will this take? For every read, what do I have to do?
Check 3 billion.
Check 3 billion, right? For every read, right? And then within each read, I have to match every position, right? Okay, so here's actually an example. So let's say my genome length is n. I have m reads to map, okay? The reads are length L. The number of mismatches I allow is D, okay? And let's say I have a computer that can compare a match to a mismatch 1 million per second. I don't know if that's fast or not, but let's just say that that's the case, okay? So then, so it would take me n times m times L times t seconds, which is like, I don't know, 27 something seconds, or 864,000 years, okay? So that's obviously not a great algorithm, right? So what are ways to improve it? So what are some obvious things you can do? Maybe not solve it completely, but improve the algorithm. Yeah?
When they check a fraction of the string, and it doesn't meet another threshold that's divided by that fraction, then just quit before you check the whole thing.
Yeah, so you can, instead of checking all 30, you can like maybe check like the first 10 and then see if it. So maybe that, let's say that saves you a factor of 3 or something like that. You know, let's say it saves you a factor of 4. Okay, that'd be optimistic. Then that'll maybe reduce it down to 200,000 years. So, but you've saved 600,000 years, right? So you look positively, right? Any other ideas? Yeah. Any other ideas? Yeah. Yeah, you could do a hash, but hashes are complicated because hashes only match exact sequences. But here you're allowing for errors. So, but before we get to that, are there any other kind of simpler? Yeah. Well, yeah, you can run in parallel. So that means that if you have 864,000 computers and you each run them for 1 year, then it'll work too. Yeah. Yeah, there are. But yeah, we'll have to talk about how do we do that. That's exactly the kind of the idea. But what are other kind of straightforward things you can do? There's ways to save least factors of 2 pretty easily. So do you have to check when you're going along, if you find it, do you have to keep going? No, so that'll save you about half because you usually, yeah?
How do you know if you find it and it's optimizing for the minimum edit distance?
Oh, so no, the problem is defined that you have a minimum threshold. So you're saying that like within any time if it's within 2. Yeah, finding like edit distance, that's even harder because of alignment. We'll talk about that later maybe. Yeah? Yeah, so you can basically pre-process your genome to find, like if you have any exact matches, so that might save you like 10%. So that saves another 80,000 years, but you're still, yeah. Okay, so now let's go back to this hashing idea. How do you do it with a hash? So everyone remembers hashes, right? So hashes are great. For example, if you had to find an exact match of 30, that would be pretty easy, right? What would you do? You just build a hash table, right, where the key would be the sequence length 30. The value would be what? The position in the sequence, right? And you just look it up, okay? But problem is that you want to have a hashing, well, let's say in this case with 2 mismatches. So how would you do that? Any ideas? Yeah?
You can take the read and just generate a list of the possible.
Yeah. So one thing you can do is you can actually, instead of looking up the read, you can look up the read all 2 like distance away from it. That's 30 choose 2 times, for like 30 choose 2 times 4 to the 2. So it's 16 times 30 choose 2. It's going to take you a long time. Yeah.
Get rid of the 3 billion.
Yeah. You get rid of the 3 billion part, but it still takes you a long time. Yeah. For every read, yeah. So, okay, so I'll give you guys, so I'll tell you how to do it. So the key observation, and maybe you guys can figure this, is that most positions of the genome match very poorly, okay? So we're kind of wasting our time most of the time. And we're looking for a very few number of mismatches, okay? So the algorithm that we talked about just now, if you're looking for a position that matches only 5 out of 30 places, it would still work, right? So whenever you realize you're solving a harder problem version of the algorithm than you have to, that gives you an idea of where you think you can improve it. Okay, so here maybe we can take advantage of come up with an algorithm that only works when d is small, right? And so the key, and I want to point out because of this, part of our read will match exactly. Okay, right? If you have only, most of the read matches exactly. So any ideas on how to take that into account? Yeah.
Hash it into 10 sections.
Yeah. So that's basically the idea. So here, let's say, what does it mean for a substring of the read to match exactly? So let's say I'm only lagging 2 mismatches. So what I do is let's say I split my read into basically d plus 1, in this case 2, like equal parts. Okay. So what's going to happen? Where can the mismatches go? Okay. They have to be somewhere in the read, right? So what I'm saying is that there are 3 kind of worst possible cases for the placement of the mismatches. Okay. And this is what I mean. These are the worst possible cases. 1/3, they fall into different thirds. Okay. But what happens in each of these cases? 1/3 is what? Huh? 1/3 matches perfectly. So here, this third matches perfectly. This one, right? Why are these worse? What's a better possible scenario? Yeah, both in the same. Then you have 2 that match perfectly. Okay. So so there's always going to be a perfect match of L/3. So then what could we do? So think of hashing, right? What can you do? So yeah, now it's just combining ideas that already people have mentioned. Yeah, any ideas?
Make a hash out of each of those thirds and then compare it to a hash of the original.
Well, not even a hash. You can, let's start with just the first thing you said. Just hash. So you can basically make a index, right, like a phone book or index or hash table for all sequences of length 1/3, L/3. Okay, so here is L/3. You know, whatever. This is all A's up to all T's. Okay. First of all, how many entries are there here? We just talked about this. 4 to the about a million, right? Okay, very good, very good. Okay, and then here would just be where they occur in the sequence. Okay, right? So then, and so if I have a table like this, how can I look it up really quickly? How do you look stuff up really quickly? Huh? Okay, how many people here, I know you guys are too young for this, but how many people have seen a phone book in their lives? Like maybe it's used as like something you stand on to get like something out of the kitchen, right? Do you remember, does anyone here know how to use a phone book? Well, it's alphabetical order, but how do you, like if you're looking something up, how do you use the phone book? Do you remember? Do you start on page 1 and flip through until you find what you're looking for? Huh? Yeah. What you do is you like look in the middle, right? And then based on the alphabetical order, you like flip halfway between, not exactly halfway. We're not so type A, but you get the idea, right? So essentially with that, if you always like search in the middle and kind of go like that, you can very quickly find the entry that you're interested in, right? Okay. So first of all, okay, so now if there's 3,000, if there's 1 million of these guys here, okay, there's 1 million of these rows, how many different values are there over here? Huh? Yeah. No. So total numbers here. How many total numbers are there on this? Like if I drew this out on the whole board, all million and then all the numbers, how many total numbers would there be? 3 billion. Yeah, right. Because every position has a sequence length 10. Okay. So then on average, how many entries per row do I have? Huh? 3,000, right? Yeah. And the formula is going to be n over 4 to the 10th, right? So you guys see how there's like this exponent in the denominator, okay? That's actually good. That's really going to help us because that's what makes it faster. Okay, so then how do I use this sequence? What's the algorithm? Now I built this, how do I use it? Okay, so I give you a read. What do you do? Okay, don't say like I take it and you slide along the genome. That's not the right answer. Okay, so I give you a read. What do you do? Huh? Well, no, but the read is length 30. This only looks up length 10. What do you do? So I give you a read at length 30. What do you guys do? You split into thirds. Okay, and so for the first third, what do? You look it up, and you get a bunch of positions. What do you do with those positions? Huh? Yeah, you check the whole read against those positions. Okay. When I'm looking in the middle third, right, I don't compare exactly those positions. I compare that position minus 10, right? Okay, but basically, this tells me only the places I need to look. Okay. And okay, so how fast is it? And I want to point out, actually, if these are length 15, then they'll be on average 3 positions. So that's something we'll get to in a second. Okay, so we look up each third. Okay, so our index contains entries of length 10. Each entry contains on average 3,000 positions. So for each position, we need to compute the number of mismatches. So the running time is going to be L, right, the size of the read, okay, times M, which is M I think is the number of reads, okay. And then for each read, it's going to be 3, because we have to do this for every third, okay, times N over 4 to the L over 3, right, which is going to be, this is basically 3,000, okay, times T. So it's going to be like 81 million seconds, which is 397 days. Okay, so, okay, it's still only 3 years, okay, but it's a lot faster. You can imagine that, you know, this kind of idea, you can even do it faster. Okay, so, but actually, interesting, if L is 45, and you're looking at 15, then it would only take like 1 day, because you have many fewer, okay? And that's intuitive. You think about it, if you're trying to find sequence of length 30 with 2 mismatches versus 45 with 2 mismatches, your sequences are actually closer, right? So that's something to think about. Okay, so so that just gives you a flavor. What are more problems? Like more problems. So for example, you need to look up each third of the read. Okay, so actually more problems. So here's actually so what if the sequencers aren't perfect? Okay, the sequencers aren't perfect. They sometimes make errors. Okay, so let me give an example. So let's say this is my genome. This is a sequence read. This position, like this read is from here, where we've been looking. This position has an error. Okay, it says an A, but it shouldn't be an A. So then what happens? If I go through the algorithm, right, I find the human genome. This position now has a new difference, right? And when I recover my sequence, I'll add in this error. Okay? So what's a way around that? Like, what do I do? How do I avoid this problem? So someone back there said it. It's a very generic, 95, like I would say 80% of the time, there's one answer to all these problems. So, huh? What do you do? Yeah? Well, no. So, no, but if you have just one read, no, that's not going to help. Nice try, though. Yeah. That's not, yeah? Well, no, but you don't know where the SNPs are. So, well, not really, because I might have new SNPs that are just unique to me. Yeah. So yeah.
Compared to a bunch of other short reads and see if that's in there.
Yeah. So basically, the answer is the generic answer. Is, if you don't know, the answer is collect more data. Okay. So if you collect more data. Here's like if I get 3 reads instead of just 1, okay, what happens? You see the true variants are in all the SNPs, right? The SNPs are in all the reads, but these errors are only in some of them, right? In only like one of them because they're independent, right? So then what I do is I can use what's called the consensus, okay? Just take the most common value. So here G is the most common, here's T is the most common, right? So sorry, here, oh, this is a really bad error. Here you made an error and you happen to be right. Okay, but this one, okay. This one here, okay, so yeah. I do this so that way for people who don't come to class and just look at the slides, it, you know, gives you guys an advantage. It's on purpose. Okay, so then you can talk about how, you know, but of course, you know, just by chance we could have only, maybe we had more than one error. If we had two, both of these had an error, then we'd be wrong. So you can think about, like, what does that mean? Like, how many examples you need to have compared to your error rate, okay, in order to do that. So that's something called coverage. So how many times do we read a certain position? That's called coverage. How much coverage do we need? Well, that's a function of your error rate. You can think of, like, let's say your error rate is E and your coverage is 3. This is basically will make an overall error if this is true, meaning that we have, this is the probability of having an error in all 3 of your reads, right? Like, or, so if you have 3 reads, remember, and you're taking the most common, you need to have, if you have only 1 error out of those 3, you'll be fine. So you need to have either 2 errors or 3 errors to have a problem. So this is the probability. This is 3 errors, okay? And then this is 2 errors. There's different ways to have that. So it's approximately 3e squared, okay? Listen, this is, you guys are all like writing this down and stuff like that. This is not, we're going to actually have an entire lecture on this, okay? I'm just trying to give you an overview of the kinds of things that we're talking about. How do we figure this stuff out? What's the math kind of behind this, okay? So another thing is that humans have 2 chromosomes, okay? Sometimes you have a read from only one chromosome. You're like, each of us has 2 chromosomes. So half of the reads on average come from one chromosome, half the other. What if there's a variant in only one of them? That also causes lots of complicated. Okay, here's another one. This is actually a good project. This is really hard to solve. Let's say in my genome, what happens is I have this A that's an inserted. So my whole genome is shifted over by 1. Okay, I have an extra, instead of you guys, all your genomes are length 3 billion, mine is length 3 billion and 1, because I have this extra A here. Okay, there's a lot of insertions in the genome. How does that affect? So if I have a read that covers that insertion, what happens is that this algorithm that I've had doesn't work really well. If I line it up like this right after the read, right, all of these positions mismatch, but all of these match. If I move it over by 1, these positions now match and all of this mismatches, right? So this is like not a random structure. So how do I deal with this, right? Like, I can try to use edit distance or something like that, but it's very complicated. I mean, one thing I can possibly do is just consider all possible kind of placing gaps in the human genome and then running my algorithm, okay? And then it would work because this matches really well. But obviously, how do I do that? So that's a good final project, how to do that. Okay, so yeah, so this is difficulty. You have to use alignment, you have to edit distance. This is not an easy problem. Okay, so there's many other challenges. So there's repeated regions of the genome, coverage of sequences is not uniform, and also there's large memory requirements. Okay, so let's just think about just that hash table we had. If you're just storing, you have to store 3 billion integers. How much does an integer take? How much space does an integer take? 32 bits, right? So if you have 3 billion of them, how many? So let's just make it simpler. It's 4 bytes, okay? If you have 3 billion of them, a billion is what? A billion bytes is what? Huh? Oh, no, yeah, so a billion bytes is a gigabyte, right? 3 billion times 4 bytes, it's 12 gigabytes, right? Just for that index, okay? So that's problematic, okay, right? So that's how do we do that? So actually one of the lectures in class, I'll tell you, is that there's actually a way to do that, to have it be like 600 megabytes, the whole thing, but without using this this kind of hash that's designed for sequences and works really well for genomes, and you can do it with less than a megabyte. So these are the kinds of things. These are all kind of computational problems that we're talking about. And so that's going to be, yeah, so that's it. That's all I have. Okay, so I want to just mention that these are the kinds of sequencing problems, and then we'll also have another whole set of projects that are related to association, which are more statistical. It might be a little bit easier for the more students from the biology side that don't have a lot of programming. Okay, great. I'll see you guys on Wednesday, and enjoy the videos.